So this is Alex Pickering. So he's going to be giving the majority of the presentation and the walkthrough. He's our technical expert on Cellenics, and he is the primary technical support person for our HMS instance of Cellenics. So any HMS users, if you have issues with your account or you need help getting started, Alex is the person to reach out to. So we have a pretty interactive workshop planned for you today. We'll have a brief introduction by Peter Karchenko, who's here, who's now is at Altos Labs in San Diego, but was a professor at DVMI for a number of years, and this is kind of his brainchild, if you will, and like initial efforts. So welcome everybody, and without further ado, we'll turn it over to Peter.
Thank you. Can I share a screen then? Thanks. Disabled participant screen sharing. Can you add me as a co-host, please? Thank you. Yeah, so I mean, I'll just say a few words and kind of quick overview of some of the basic steps maybe that. But the general idea is that the single-cell data presents a number of computational challenges for analysis. It's sparse, it's noisy, it can get quite large, and so there have been a number of tricks developed along the way for how to handle it, how to normalize it, how to get sort of stable results out of it, how to accentuate the differences that are within this data, or the patterns within this data. And these methods are generally available in this platform. They're implemented there. But I just wanted to kind of give a quick overview of maybe what they are, just to give you a bit of an intuition for them. So in general, single-cell experiments are very nice for studying complex tissues. This is a snapshot of mouse blood, for instance, and you're seeing not just distinct subpopulations like macrophages, but can also trace trajectories here and get a sense maybe for the dynamics of the tissue. And this is a single kind of shot experiment, so it can be quite powerful. I think the nice thing and the challenge of the single cell data is how much has been progressing. So here on the right, just a quick plot, as a function of time, comparing the progress and just the number of cells that are being measured by different experiments, different assays. Comparing it with a Moore's law, which is a number of transistors and processors. And it's been growing very rapidly, and you can see that it's sort of growing faster than Moore's law in a way. And what that means is that things are more difficult to compute every year, effectively. So the numbers of cells are growing, the numbers of experiments people can do independent, let's say, conditions or replicates is growing as well. There are kind of clinically oriented studies with many patients, for instance, being measured. There are also consortium scale efforts to create atlases, and those can contain millions upon millions of cells. All that brings up computational tasks as well as some statistical questions. And so the platform, I think, is meant to wrap around some of these computational issues to give a more user-friendly interface to analysis, or at least some of the standardized analysis. Yeah, so I'll spend, I think 10 minutes or so just going through some of the basic steps in the analysis. And much of it pertains to individual data sets, how we analyze individual single-cell RNA-seq data sets, and I'll touch upon integration just a bit. So there are certain steps that are kind of necessary to do this processing, to even get the data into a shape where it can be interpreted. Most of the assays these days include unique molecular identifiers, which are little barcodes on individual cDNA molecules. And so, for instance, one of the first steps you have to do is just to sort out barcodes. The both cellular barcodes and these molecular barcodes both need to be counted, and the reads need to be effectively classified into molecules and assigned to particular cells and particular genes. And so there's a number of tools that have been developed to do this. Cell Ranger is probably most popular one because most people use 10x assays these days, and CellRanger is a default for that. There are other tools as well, the open source ones that have been developed. And so they all make some assumptions, and they all have some approximations and tricks for let's say accounting for sequencing errors in the barcodes. So here's a quick illustration of one method, for instance, for accounting for sequencing errors is to do a little clustering. So if you, let's say, counting unique molecular identifiers for a given gene. So you encounter a number of reads, and many of these reads have this particular barcode, but then some of the reads have a barcode that differs by one letter, right? And so this particular heuristic algorithm connects the barcodes that differ by one letter by an edge, and then at the end, after you've gone through all the reads, for instance, in this library, you simply look for connected clusters like this, and you collapse them into a one molecule, right? So this is just likely to be a sequencing error, so we will treat it, all these reads, just as one molecule. So a number of heuristics like this built in, and they work up to extent. So for instance, if you have a lot of sequencing errors, that will lead to overcounting, let's say, of molecules. Very important step is filtering. And again, this is where a lot of heuristics are used. So in these, let's say, droplet-based methods, you'll encounter droplets that don't have cells. Actually, quite a few of them don't have cells. And they're not entirely empty. They will have a little bit of a background molecules in them. Sometimes it's just ambient kind of mRNA background that was more or less evenly spread out throughout the solution. Sometimes it can be sort of a residual, a comet sort of tail from a cell that was going in the channel right before this empty droplet, right? So it can actually be quite specific and not, similar to the ambient background. The most basic way in which people filter them, but quite effective, is simply looking at how many molecules you have. So effectively just discarding a tail of this kind of size distribution. This is number of molecules in a cell, and just discarding it, thinking that it's mostly represents empty droplets. You can also try to classify empty droplets, and some of the methods again do this using more sophisticated classification methods. The other part here that is becoming increasingly important is recognizing doublet populations. So if you imagine, we do an experiment and we sequence this 3 major cell populations, red, green, and blue. But let's say some of these droplets capture pairs.
Sorry, Peter, you're cutting out a bit.
Sorry, I'm not sure why. Is this okay now?
I can hear you just fine, Peter. Maybe it's on Jacqueline's end.
Okay, so yeah, so sometimes droplet will capture both green and a blue cell, let's say, and sometimes both red and a blue cell, and so these cells, these doublet, droplets, will show up as cells with a different transcriptional signature, and so if there are enough of them, they can even form their own cluster. And so these are important to recognize because they're artifacts, right? There's no such subpopulation by itself. And there are a number of tools that are being used to recognize or classify these potential droplets. Again, they're built on their own assumptions, and they work up to extent, right? So if you run doublet detection, which is a prudent thing to do, on a diverse set of populations here, it will probably give you pretty reasonable results. It will detect these very clearly mixed populations, and you can remove them. But if you run a doublet detection on a closely related set of cells, let's say just the red cluster on its own, it might give you some false positives, or it will, at the very least, filter out some of the cells that are probably just fine, just red cells. So a lot of analysis for individual data sets just have to do with normalization of expression estimates. And really, there are two parts to it. There's accounting for how deeply the cell was sequenced. This is cell size estimation. And the other part is accounting for variants, basically, of different genes. And I'm not sure if I'll have an illustration of this, but yeah, a little bit. So there's a kind of a key statistical property, that if you look at the mean expression of each gene, every point is a gene here, and variance. So the variance will grow. The higher is the mean expression, the higher is the variance. So if you simply take most variable genes with the highest variance, you'll simply be taking highest expressed genes. And those are not necessarily most informative. You might have a structural, let's say, protein and a transcript for it that's just simply expressed at high levels, but it doesn't actually distinguish subpopulations. So we typically want, because we want to highlight subpopulations, we want to focus on genes that are variable more than one would expect from this general kind of dependency. So here's an example. This is, again, a real data set, a PBMC data set, and we have observed mean, observed variance. And if we just take a look at these red genes, they're the ones that vary more than expected. And so if we just look at examples of some of these genes, and we see that they nicely mark particular subpopulations within this experiment. And distinguishing subpopulations is kind of a key aim of the analysis in single cell data. So focusing on this set of overly variable genes relative to their general dependency on the mean is a useful technique. Yeah. So and then you can go into further analysis, but a key aim, as I mentioned, is distinguishing subpopulations or structure of subpopulations in the data. And the general technique that's being used for this is we start with dimensionality reduction, because if we just work in the space of these tens of thousands of genes, it's very difficult to actually assess which cells are similar to each other, which ones are not. It's a property of a high dimensional space. That's just mathematical property that makes it very difficult to assess. And plus, if you have the noise, then statistically it's difficult to assess, to distinguish cells that should be within the same subpopulations versus distant ones. So we deal with this by reducing the space to a lower number of dimensions. And there are many ways of doing this, and this is kind of a major area. You can use very simple techniques such as principal component analysis, sort of standard things. You can use very fancy things like these deep neural net autoencoders that will effectively try to find optimal nonlinear sort of embedding or reduction of these individual gene values into some sort of latent dimensions, right? Now, let's say a dozen of latent dimensions. Even in these latent dimensions, the structure of the populations is often very nonlinear. And so to capture it, people use graphs. And I'll give you a little example, cartoon version here. So if we simply start connecting closest points, right, the closest cells in this case, on a local level, we just for each cell find its closest 3 neighbors, for instance. And that gives us local neighborhoods, which hopefully connect cells of the same type, of the same state. But this process also works nicely on a larger scale. So if we start looking further and further, we'll see some kind of a structure emerge, and then eventually we'll see what is a toy elephant in this case. And so the elephant is a complex shape topologically, right? And it would be difficult to estimate with a sphere or something else. But a graph like this, a nearest neighbor graph, is actually very good at this. And so it effectively gives us a topological approximation. So for instance, we could distinguish that these 2 legs, the shortest way of getting from one leg to another is through the torso, not directly, right? So we have an idea about connectivity between different populations and how they flow into each other. And then the clustering that is very commonly used, trajectory analysis, and so forth, they're effectively ways of formalizing what the structure of these populations are, whether it's continuous structures or discrete structures like clusters. But there are ways of basically approximating this cloud of points in this and the structure that it takes. And that's just important to keep in mind. So they're all approximations, right? So if we're clustering this elephant, we could do this at multiple resolutions. A trunk can be clustered basically in many partition in many pieces or one piece, and both are meaningful, right? So that there's no inherent, I think, mathematical meaning to the resolution of clustering that you're doing. But obviously, in terms of interpretation, they can be very different. You might want to use coarser interpretation, for instance, grouping all the T cells in one group, if that makes sense for the biological sort of story or analysis that you're doing, versus subdividing them into very fine subpopulations if that's what's needed. The other thing to keep in mind that all these methods can give you very different or slightly different results in terms of clustering. In particular, cases where you have a cluster that can be subdivided in a sort of rather arbitrary manner, they will be unstable. So that's just something to keep in mind. The last thing I wanted to show, well, I wanted to mention integration, but you'll probably talk about this quite a bit. The main idea is that if you have multiple data sets with different sort of batch effects, right, technical effects or individual variation, you can kind of align them and bring them together into a more coherent manner. And that will allow you to basically find the corresponding subpopulations between different data sets. And with the multi-sample designs, experimental designs, that's a very important step. And there are many ways of doing this. I think Seurat aligner and Harmony are probably one of the most popular ones. Trajectories, yes, I'm not going to talk much about this, but if you have these continuous patterns, let's say in this case it's a differentiation pattern that branches and so forth, you can approximate these continuous patterns with trees or curves and other sort of simple shapes that just give you an approximation of the trajectory. But again, you have to use them in a very cautious manner. So there are many trajectory tools, but if you just let them loose on a data set, that can give you a very misleading answer, because all they do is just find a continuous trajectory. So here's a contrived example. This is a PBMC data set, and we just asked a common trajectory fitting tool to fit a trajectory connecting all the PBMCs effectively, and it will happily do this. But of course, these are all related lineages, but they're related through progenitor populations, which we don't see here. So the trajectories that will fit here may have, no actual biological interpretation. Again, these tools, they're mathematical tools for approximating the structure of the data, but you have to use them carefully to answer the questions that, actual biological questions. All right, so I think I'll end here for a quick intro.
Great. Thank you so much, Peter. Really appreciate the introduction. I'll just get my screen up here. Okay, you guys can see my slides?
Yes. Yes.
Great, thanks. So as Jacqueline mentioned, I'm Alex Pickering, and I'm a senior scientific programmer at DBMI, as well as the lead developer of Cellenics here at HMS. And I'm going to be running the workshop as well today. So the first thing that I want to say is that I really want the workshop to be as interactive as possible, given the limitation that there's one of me and many of you. So I'm going to be asking you guys to write down in the Zoom chat throughout the workshop. And if you have any questions, please post them down there, and either Jacqueline and Genoa, they'll take note of the questions, and I'll answer them during the exercises. So the first question that I have for all of you, even before we get to the question that's up here on the slide,
You accidentally muted, Alex.
Oops, you can hear me now?
Yes, we can.
Great, thanks. Sorry about that. My first question for you guys, even before we get to this one up here, is there anybody here who either did not register for the workshop or who didn't get an email from me this morning with details on how to log into Cellenics? And if that does apply to you, please let us know down in the chat, and either Genoa or Jacqueline will be able to make sure that you have those details so that you can log in. So assuming we get that sorted, my next question for all of you is how experienced are you analyzing single-cell RNA-seq? Do you have a little bit of experience? You know, are you very experienced, or is this your first time? And the reason why I ask this is just so that I can make sure that I'm pitching the workshop at the right level, and so that I'm giving you guys plenty of time to answer the exercises. Great, and I can see that a lot of you are already writing that in, so that's really great to see, super helpful. My next question for all of you, after you answer the first question, is do you have a single-cell RNA-seq data set that you're either trying to analyze right now or that you would like to analyze? This could be a publicly available data set. This could be a data set produced in your own lab or in the lab of a collaborator. So if that's you, let us know down in the Zoom chat. And the reason why I asked this one is I'll follow up with you guys directly after the workshop, just to make sure that if you need any support for Cellenics that you're getting it. And that's really one of the take-home messages for today's workshop, is that I'm your go-to person here at HMS if you need any Cellenics support. So please don't hesitate to either just book a Zoom with me or send me a quick email. I'll circulate these slides after the workshop so that you have access to these links. I'd love to hear about your project and make sure that we get your analysis going. Okay, so the first thing that we need to do for the workshop is obviously to get you guys all to log in to Cellenics. So if you could all please open, preferably a Chrome browser and go to cellenics.hms.harvard.edu. And hopefully Jacqueline or Genoa can either post that in the chat as well, just to make sure you can find it. And when you get there, you're going to see this sign-in page here. If you're, your credentials, go ahead and log in. If you're a new user, you should have received that email from me this morning giving you your login details. Check your junk folder if you can't find it. If you're an existing user, obviously just log in using your usual credentials. If you're having trouble remembering them, you can always use the forgot your password workflow, and let us know once you've managed to sign in. I see that some of you already are, which is really great to see. And if you're having any trouble finding your credentials or logging in, again, just please let us know down in the chat, and either Genoa or Jacqueline will be able to get you sorted out. So just while I give you guys another couple minutes to log in, I'm just going to show you the next couple of slides so that you know what to expect once you've managed to get into the platform. So when you first log in, you land on the data management module. Over on the left here, these are the navigation controls for the 4 modules of Cellenics, and you don't need to click on these now. We're going to be stepping through each of those during the workshop. Just to the right of that is where you can see the projects that are available to you. So if you're a new user today, you're just going to have this one COVID-19 data set that I've created and processed for you. If you're an existing user, you obviously might have other projects here as well. And if that's the case, go ahead and find this COVID-19 data set that was created last week and click it. And when you do, you'll see over here in the project details area that this particular data set, it has 3 samples, and I'll go over in the next slide what those samples are. Importantly, if you're you can access your credentials, your account settings up here in the top right, and that's just, you just want to make sure you change your password if you're going to be uploading your own data to Cellenics. We've obviously issued a lot of temporary passwords for today's workshop, so just make sure you guys do that. As far as the data set that we're working on today, it's from this paper here on COVID-19 associated MISC or MISC, which stands for Multisystem Inflammatory Syndrome in Children. As I mentioned, the data set has 3 samples. It's PBMCs that were isolated from children who were infected with COVID-19 and then subsequently developed this MISC. And 2 of the samples are from patients during the acute phase of MISC. It's patients P13 and P14. And the 3rd sample is also from that same patient P13, but it was collected at a later time point, so it was during recovery of that patient.
Alex?
Yeah.
Sorry to interrupt. We have a couple of folks who are existing users who can only see their data sets and don't see the training set, the COVID set.
Okay. Yeah, maybe in that case, I mean, try searching for it up here if you can. If you don't see it there, I might have goofed up something, and maybe either you and Genoa could give them some of the credentials for the burner accounts, so that they can access the data set.
Yes, so those of you who've reached out to us will send you a burner account to use for today.
Great, thanks, Jacqueline. So as far as the tissue type, the PBMCs, I suspect that most of you are familiar with this tissue type. So just very high level, this is an immune cell fraction that's isolated from the peripheral blood. So it has, various immune cell types in it. And they're widely used both because they're, relatively easy to isolate, as well as they're readily accessible in human subjects. As far as the disease itself, MISC, you don't really need to know much about it. So just for some context, it usually develops about 4 to 6 weeks after COVID-19 infection in children. It's characterized by a marked inflammatory state in various organs, and it can be quite serious, particularly because of the cardiac symptoms that can develop. And the authors of the paper essentially state that, our understanding of this disease is fairly poor, and so they want to understand how the immune system changes during MISC. So hopefully most of you have managed to log into Cellenics by now. Again, if you're having trouble let us know down in the chat. You still have a few minutes, and Jacqueline and Jenna will be able to get you sorted out. Once you do log in and you're into data management, I have an action for all of you, and that's to go and click the Go to Data Processing button. Again, making sure that you have the COVID-19 data set selected. And when you do, you're going to land on the data processing module. And it's worthwhile me just giving a very high-level overview of exactly what it does. So this is a 7-step sequential pipeline. It's already being run for your COVID-19 data set today. The first 5 steps are filters. So these are removing empty droplets, dead cells, doublets, etc. After each of your samples is filtered, the next step is to integrate those samples together. And this is also where that PCA dimensionality reduction is performed. And then that reduction gets used as an input to the last step, where either a UMAP or a TSNE embedding is generated, as well as the automated clustering is performed. And there's lots of settings that you can change for all these steps. Just to note, for the first 5 steps, if you need to change a setting, you first need to click the manual toggle before you can adjust the threshold. And my only request for today is please just leave the settings as they are, and don't rerun data processing. And that just makes sure that we're all looking at the same UMAP, as well as you're not waiting for data processing to rerun. It does take a few minutes to run, and I don't want you guys to waste your time. As far as what the data processing module looks like, so I mentioned that there's 7 steps. You can navigate between them either using the drop down up here in the top left, or using the navigation arrows up in the top right. We have tried to make the platform as biologist friendly as possible. So there's a number of information tooltips throughout the platform. And these will give you details on, exactly what why a particular step is important, what kind of methods are being used for that particular step. And they'll often link to additional materials about those methods. So definitely do check those out. They can be really good for understanding exactly what's happening to your data at each step. Finally, in the top right here, this is just the pipeline status indicator. So for all of you, this should hopefully look like it does up here, just indicating that the pipeline is complete. And this will obviously, update itself if you're running a new data set. It'll to indicate exactly what's being run currently. So now that you have sort of a very general sense of what the data processing module looks like, I'm going to jump into Cellenics just to take you through each of the steps and give you more details on each of them. So the particular data set that I'm going to use for that is just it has 2 samples. It's just for demo purposes. And the first step that we land on is the classifier filter. And the goal of this step is to remove droplets that don't have any cells in them. So these are the empty droplets. And the way that this filter works is it essentially runs a hypothesis test for each droplet, asking whether it's empty or not. And so you get a p-value, you adjust it for multiple hypothesis testing, you end up with an FDR, and then any droplets that have an FDR higher than the threshold here will be discarded from your sample. And shown here is just a knee plot, and we can see that the orange and the gray those are a mixture, some of the gray based on the FDR. Those are the ones that are being discarded from this particular sample. And just for those of you that might not be familiar with it, the knee plot, this is plotting the number of UMIs, which stands for unique molecular identifiers. And this is essentially, it's the number of reads that are mapping to individual mRNAs in your droplet. And so it's essentially the counts in the droplet, and it's just removing things like PCR duplicates, various technical artifacts.
Alex, sorry to interrupt. I'm not sure. So I'm on one of the burner accounts, and my graph looks different from yours. I don't have a gray bar at all.
Oh, yeah. No worries, Jacqueline. So good point for me to make. So this data set that I'm looking at here is not the workshop data set. I'm just showing you guys an example data set, just to give you a demo of the data processing.
So this has already happened on the data set that we're looking at.
Yeah, and the COVID-19 data set is not this one, and it'll look different than the one I'm looking at.
Okay.
So the next filter is the cell size distribution filter. This filter is also for removing empty droplets. And the distinction between this filter and the previous one is here you can set a hard threshold, this red line, for the minimum number of UMIs that you want to allow in your droplets. And because this filter overlaps in functionality with the previous filter, this filter is disabled by default, but you can enable it by clicking this button up here. And sometimes it will make sense to do so. The previous filter, the classifier filter, it can be overly conservative, so not filtering out enough. So if you find later on in your data exploration that you have, a group of cells that's really only distinct in terms of having low UMIs, you might want to consider coming back and enabling this filter and adjusting your threshold. And you'll see later on exactly where you can see that. The next filter, step number 3, is the mitochondrial content filter. And this step is taking a standard approach to filter out dead and dying cells. So what we're looking at here is a histogram of the percentage of mitochondrial reads in our droplet. So this is the percentage of reads that map to one of the mitochondrial genes. And then everything above any droplets that have a value higher than this red threshold are discarded from the sample. The next filter is the genes versus UMI filter. And what you're looking at here is a plot on the y-axis. This is the gene counts. So this is the total number of genes that have at least one read in them. And that's being plotted as a function of the total number of reads in a droplet. So you expect to see what's shown up here, where with increasing sequencing depth, so a higher number of total reads in a droplet, you also detect more and more genes in that droplet. And then anything outside of these red threshold lines is discarded from your sample. And this is one filter where if you do see a substantial number of cells outside of these red threshold lines, it's worthwhile investigating exactly what those cells are, and then determining at that point whether or not it's appropriate to filter them out. And the most common scenario that you'll see, and you'll see this today, is a group of cells down here. And you can think of these as low complexity cells. So for a given sequencing depth on the x-axis, there's relatively few genes that are detected. So these cells are pumping out a bunch of mRNA, but they're just for a relatively small number of genes. The fifth and last filter step is the doublet filter. So as Peter mentioned, there's sort of three cases that can occur for the droplets that we sequence. The first case is that a droplet is empty. There's no cell in it, and we've already removed those from our sample. The second case is that there's exactly one cell in our droplet, and this is the ideal case. These are the droplets that we want to analyze. And then the third case is that there's two or more droplets or cells in our droplet, and we want to get rid of those as well. And so that's what this filter is doing. And the way it does this is it essentially trains a machine learning model in order to predict a probability for each droplet as being a doublet, as well as a threshold to use for classification. So any droplets that have a predicted probability higher than this red threshold line are being discarded from your sample. The next step is data integration. So we finished filtering out our samples, and now we need to combine our data. There are a number of methods available for doing this. There's sort of standard methods that you can select from here. As Peter mentioned, the goal of these methods is to sort of eliminate these, sample to sample batch effects. And you can adjust the settings for the methods as well as for the PCA reduction that occurs on your data set after. This step also has a couple of plots that you can use just to evaluate the quality of your integration. So the one you're looking at up here is it's being colored by sample identity. This particular data set has two samples. And on our embedding, we can see that the samples are superimposed on each other. So that's what we want to see. The other way we can look at this is as a frequency plot. So down here, we have all of our automated clusters, and we can see that each of our samples has cells contributing to all of those clusters. And if you don't see this for your data set, you want there to be some biological explanation for it, like maybe a particular group of samples, you don't expect there to be a certain cell type. And if you can't come up with that explanation, that's where you might want to consider trying out a different method. Okay, next step is where you can configure your embedding. So you can select either a UMAP or a TSNE embedding, as well as the settings for that. And this is also where you can adjust your clustering settings. So right now it's just Louvain. And as Peter mentioned, this is where you can adjust your resolution as well. So if you increase it, you're going to get more clusters. And if you decrease it, you're going to get fewer clusters. There's also a number of quality control plots available to you in this step. So you can plot on your embedding the percentage of mitochondrial reads, your doublet scores, as well as your number of UMIs. So those can be useful as well for evaluating the quality of your data set. Great. So now that I've given you sort of all the details of data processing, we're going to jump into the first exercise for today. So in this exercise, we're going to look at the three samples from the COVID-19 data set, and we're going to look at whether they differ in terms of quality. So we're going to look at the proportion of dead cells in these samples, the proportion of doublets, as well as whether the samples are well integrated. So in order to do this, you're just going to go to the data processing module. You're going to navigate through each of the steps and then pay particular attention to the mitochondrial content filter, step number 3, the doublet filter, as well as the integration step. When you finish, please type done one in the chat, just so that I can make sure that you're all given enough time for the exercise. This should take you guys about 5 minutes, and feel free to take a 5-minute break after as well. And I'll answer any questions if any have come up as well. I can see that some of you guys are putting done one down in the chat. Really great to see. Really appreciate it. You have about another minute for the exercise, and please give yourself a 5-minute break once you finish as well. So, um, just in case somebody's just mentioning to me that they're having trouble seeing um, um, a plot in step number 7. So just in case any of you are also having trouble seeing this plot, one thing, there is a bug that sometimes pops up. So one thing you can try to do to resolve it is just to come down to the clustering settings. And if you just change your resolution to something else like 0.9, let it recalculate the clusters, and then change it back to the 0.8 for the workshop. And hopefully that will resolve it. Okay, so I saw a lot of done one in the chat, and hopefully you had a chance to take a little break as well. Can you guys see my my screen up here?
Yep.
Okay, great. So we'll jump into the answer for this first exercise. Before we do, it's worthwhile just reflecting on the purpose of this exercise. A relatively common question that we'll get is, you know, a user will get in touch and say, I have a sample that I think might be, you know, lower quality than the other samples in my data set. Do you think that I should exclude it from my analysis? And that's really the goal of this exercise, is try to give you the tools so that you can evaluate that. So the first thing that we were looking at is the proportion of dead cells in our sample. So this is the mitochondrial content filter, step number 3. And the first thing that stands out is that this sample P13 acute, the plot for it looks different than the other two. But if we look a bit closer, we can see that this is because of a scaling issue in the y-axis. So it goes up to 10% here and 5% in the case of the other two samples. And this is mostly because there's just a thin green line with, you know, there's a substantial number of cells with a low mitochondrial percentage. If we look at the actual distributions themselves, they all have a median of about 7%, and about, you know, 5 or 4% of our cells have that median value. So the distributions actually look quite similar. And in terms of the percentage of cells that are being filtered out, they're also, you know, all around 10%. So nothing concerns me here. One thing that you would want to look out for would be a rightwards shift in one of these plots. So if one of your samples had on average a much higher mitochondrial percentage, that might indicate a potential quality issue with that sample. So the next thing that we looked at for this question is the doublet filter. Again, this P13 acute sample, it looks different than these other two. Again, there's a scaling thing going on with the y-axis. It's, you know, very small values here and much larger over here. So we're essentially missing, you know, this high frequency, low probability droplets. And what I think is going on for this sample is that there's just a relatively small number of cells for this sample. So as you might remember, this step is training a machine learning model in order to come up with a probability for each droplet. And so I think that there's essentially just less training data for this sample, and that's why we're seeing generally lower confidence in the predicted probabilities. The other thing that we can look at is we can look at our doublet scores on our embedding, and we don't see any, you know, clusters of cells that have high doublet scores. So I'm not particularly concerned at this point. The last thing that we looked at is whether the 3 samples are well integrated. And there's a couple different ways that you can approach this. One is by looking at the embedding, where we can see that there's, you know, reasonable superposition of our 3 samples in this data set. Our frequency plot tells the same story. where all of our clusters have cells from each of our 3 samples. So the data set seems reasonably well integrated. So at this point, we're fairly comfortable with the quality of the data set. So let's see if we can come up with something sort of interesting biologically about this data set to motivate our next exercises.
So Alex, sorry, there was a question about the thresholds. Why
Sure.
were the thresholds different? Did you do that intentionally for teaching purposes or?
The threshold. So, like someone's thresholds were different than the ones shown up here, or?
Well, so you pointed out that
Yeah.
sample P13 is the scale on the y-axis is different.
Yeah.
That intentional
But-
or yeah.
Yeah, the threshold for the mitochondrial content, the percentage here is 25, and in the other 2 samples, well, one is 20 and the other is 10. I was wondering
Sure.
if,
Yeah.
why, how to make that decision on where to put the threshold for the mitochondria.
Absolutely. So usually you don't need to decide. Usually the default's pretty good. But what it essentially is doing here is it's just removing outliers from this plot. So it's looking 3 standard deviations above the median, and then it's discarding anything above that. That's how the actual threshold is calculated. So you can see it's just, there's this distribution here, and then there's this long tail of outliers, and it's just removing those. So, I mean, in the past, people did use, various heuristics where they would say, I'm just going to set it at 15 for every sample. But usually you can be comfortable with the defaults that come. Another thing that you can look at is you can look at the mitochondrial percentage, the plot on your embedding. And you can see if there's, a cluster of cells with a very high mitochondrial percentage. And if you see that, then that might be reason to come back and change one of these thresholds to try and filter out more of those cells. So hopefully that answered that question. Great question, by the way. Thank you. And if you guys have more questions, again, please don't hesitate to post them down in the chat. The thing that I first noticed about this data set when I looked at it is that 2 of the samples are from the same patient, patient 13. One when they're in the acute phase of MISC, and then one after they've recovered. And for a lot of you, just to note, the colors are going to be different. The sample colors are going to be different for you. So don't be concerned about that. So given that 2 of the samples are from the same patient, it seemed reasonable for me to see if there's any clusters that increase in proportion as patient 13 recovers. And these were the 3 clusters that best fit that criteria for me. So low in blue and lots in yellow.
Alex, sorry, there's a question in the chat. Can you just back up for a second and explain where you got this graph from, like which step?
Sure. Yeah, so this is just from data integration and going on the frequency plot. Different data set, but yeah, so just looking at the frequency plot on step number 6. So I wanted to know what are these 3 clusters? So to answer that, we're going to use the data exploration module. And so this is going to be where we find out, what's distinct about a particular cluster. These are called marker genes. What are the marker genes for a cluster? This is also going to be where you run differential expression analyses. So that means you're running a comparison between two groups of samples. So think, treated versus control samples, and you're doing it in a particular group of cells. That's, one of the promises of single cell RNA-seq. And then after you get your differential expression results, then you can run a pathway analysis on those results. So you're going to do that here as well. And so this is really where you're going to be digging into your data, looking for interesting things about it, and really where you're going to be spending the majority of the time in the platform. So this slide here is just a reference for the next exercise of the common cell types in a PBMC data set, like the one we're working on today. So I'll probably put this up partway through the next exercise, and you can choose whether you want to look at it or struggle without it. Here is just our embedding for the COVID-19 data set, and I've just gone ahead and labeled the 3 clusters that went up in proportion with in patient 13 as they recovered. And I'm gonna show you how to annotate one of the clusters, and then you guys are going to do the other 2 for the exercise. So I'm going to jump into back to Cellenics. I'm going to go to data exploration. Up here on the left, we have the embedding again. We can see our cluster 12 that I'm going to annotate. Just to the right of that, we have our cell sets and metadata tile. This is where we can see all of our automated clusters. We can select them, see how many cells we've selected. This is also where you're going to have any custom cell sets that you define, which I'll show you some ways you can do that. And anywhere that you see one of these little eyes, if you click it, it's going to color it on your embedding. So if I click my custom cell sets, you can see I've used them to draw, a beautiful chicken in what I thought looked like a drumstick-shaped group of cells over here. This is also where you'll find any sample level metadata that you've defined. So that will be in data management. I'll show you how to do that at the end. For this data set, we haven't defined any sample groupings, so we just have our samples available. We can plot those on our embedding. This is also under this Annotate Clusters tab. This is where you can access automated cell type annotation. So this uses SC type. You need to select the tissue type of your data set, as well as the species, either human or mouse, is currently supported. And my general view on automated cell type annotation is that it can be a really good starting point for your annotation, but that you still want to know how to manually annotate your data set. And that way you can just confirm the annotations that it comes up with, just to make sure that you're comfortable with them, because they won't always be right. They'll often be wrong. So that's what I'm going to be focusing on today is how to do that, those manual annotations. Over in the far right here is the genes tile, which defaults to our list of genes in the data set. Again, if we click the eye beside one of the genes, it will plot the expression of it up in our embedding. This is also where you access the differential expression tab. This first radio button here, you're going to use that when you want to find marker genes to see what is distinct about a particular cluster. The second radio button, you're going to select that one when you want to perform these between group comparisons, so treatment versus control in a single group of cells. Finally, down here in the bottom is the heat map tile, which defaults to the top few marker genes for all of our automated clusters. And you can customize the heat map quite a bit. You can change the settings and change the genes that are shown on it. And you guys will do that for the last exercise today. So I mentioned that we're going to annotate this.
In the heat map, what, like, is there a color reference for the expression? Like, what is yellow and what is purple?
Yeah, so yellow is high expression and purple is low expression.
Okay.
Good question. Yeah, it's not totally clear. There isn't a legend for this one. So I'm going to show you guys how to approach annotating this cluster 12 here. So a couple different ways we can do that. One is just a quick and dirty way. We can go to our heat map, come over to cluster 12, and then head down to the block of genes that is yellow for that cluster. So high expression. We can see here we have HBB, HBA2. If we don't know what those are, we can come up here and search for them. We can plot the expression by clicking the eye, see yes, it's mostly expressed in that cluster 12. If we don't know what it is, and we can click the name of the gene, and this will take us to the gene cards entry for the gene, where we can see that this particular gene is a hemoglobin subunit. So, likely that these are red blood cells. If we don't have enough marker genes down here, that's where you where you want to go up to the differential expression tab so that you can get the full list of marker genes. So again, you're going to select this first radio button for marker genes.
Alex, if folks, so there's a question in the chat about accidentally clicking the X on the heat map. How do we bring that back up if we've?
You might be able to do the add heat map, although I think this might be broken. So if that doesn't work, I would just refresh the page and it should be back. Great, good to see it worked. We want to get the marker genes for a cluster. So we select that first radio button. We're going to select our cluster 12 that we're interested in. And in this case, we're going to compare it to all other cells. Sometimes you're going to want to compare to just one other cluster. This is particularly useful if you're trying to compare two closely related clusters to see what's distinct about them. And in this case, we're going to keep all of our samples for the comparison. We run that, and now we have our full list of marker genes. Again, we can plot them over on our embedding, see that it's mostly expressed there. We can go see what it is. In this case, 5' aminolevulinate synthase 2, a little bit more obscure. If we read a bit further, we can see that this gene catalyzes the first step in heme biosynthesis. So consistent with these being red blood cells. So now that we know what this cluster is, we want to record that somehow. So the most common thing you'll do is just come over to your Louvain clusters, go down to your cluster 12, and click this little edit button. And then you can just change the name of the cluster. Another thing that I'll show you how to do, just so that you know how to do it, is you can come over to the embedding, click this little lasso button, and then click and drag around a group of cells. And again, you can give it a name, and that's going to pop it down into your custom cell set. So you can see here that it's, this group of cells has been added to, my beautiful drawing of a chicken. Great. So that's everything I want to show you for data exploration. What I'll get you guys to do for the next exercise is to annotate these two other clusters that went up in expression in patient, or went up in abundance in patient 13 as they recovered. So for this exercise, I'm going to steer you for the all of cluster 7 up here. I'm going to steer you to use the quick and dirty approach by just hovering over these genes in the heat map and maybe searching for them up here. Whereas for the blue cluster 4, I'm going to recommend that you use the differential expression tab, just so that you can get a little bit of hands-on experience using that as well. When you finished, please type done to in the chat. If you have some time after you finish, one thing you can do is look and try to figure out some things that distinguish this all of cluster 7 that you've just annotated from the nearby yellow cluster 2. And please give me a big done to in the chat when you finish annotating the first 2 clusters. And I'll give you guys maybe about 10 minutes for this. We have a question in the chat. Maximilian is asking if there's a way to create a new project out of one identified cluster. So great question. You definitely can do that. So the way that you do that is, say, for example, I wanted to create a new data set out of these 3 clusters, the purple, blue, and pink one up here. I would select them. So cluster 3, cluster 4, cluster 8. And then this button up here, the first one, subset selected cell sets to a new project. If you click that, it will create a new project for you that just includes those cells. And that can be a useful thing if you want to really explore, you think there might be subclusters within a group of cells. And so by subsetting the data set, your PCA reduction and your embedding, it's all going to be based on genes that have that high variability just within this group of cells. So sometimes you can pick out sort of additional subsets within that group. So great question. So we have another question in the chat. So Stephanie Moore is asking, when the data are for non-human model species, will genes still link to an external database of gene info? So I think what she's asking is like if you click the name of the gene cards, so will it still show you the information on that gene if it's non-human? I don't believe it will. I think gene cards is very human centric. I think it probably still will go to the right place if it's mouse or rat. They usually use the same gene identifiers. They're just title case instead of all caps. So they'll still give you the information for that, but you might have variable luck for other species. We have another question in the chat. Martin is asking, can we mathematically justify selection of a group of cells as a new cluster? I mean, when is it okay to manually create a new cluster? So good question, a challenging one for me to answer. I'm not sure that I can I'm not sure that I can give you a mathematical justification for when it's reasonable to sort of create your own custom cluster. I think as with anything, you want to exercise caution and make sure that you have a reasonable justification. So I think one thing that I'll often see in a data set where I'll use sort of that manual selection is sometimes you'll get things like, you have this yellow cluster here, and maybe there's some set of cells out here on your embedding that also shows up as yellow. They're part of the same cluster, and they're different. They're out here. Why are they out there? And so I think that that's a good use case where you could, use that manual selection, grab a set of cells, and in order to create a custom cell set so that you can try and figure out, why are those cells distinct from the yellow cluster? I think as Peter mentioned, there is no one correct clustering. You can change the resolution of your clustering. You can, get a ton of clusters for your data set. I think you sort of, it's sort of a more heuristic based thing at this moment. But yeah, great question. Another question in the chat. Kiong, sorry if I'm mispronouncing that, is asking how to find each closest top expressed genes. So let me just share my screen again. I sort of lost my connection there. So the question is, how do we find the closest top expressed gene? So I think, correct me if I'm wrong, but I think your question is, how do we find, the marker genes for a cluster? So again, this is where you go to differential expression. We want to say you're annotating cluster 7 up here. We're going to select that first radio button. Select cluster 7 compared to all other cells, keeping all of our samples. We run that, and we'll get a set of marker genes for that cluster 7. And then you can plot the expression of those, confirm that they are indeed high in that cluster. Yeah, and then there's various ways that you can, there's various statistics associated with those marker genes. So AUC is, a common one for sort of saying, you can sort them by AUC to see how well they classify, that particular cluster. But oftentimes it's very manual. Just, check it and see, is it well expressed in that cluster? Yes, it's well expressed in that cluster, but, it's also expressed in the nearby cluster too. So, it's probably a general marker for both of those clusters. Okay, so that's 10 minutes for the exercise. I did see a couple of done twos in the chat. I'm not sure if I might have missed some while I got dropped there, but hopefully you were able to get through that exercise. I see some more coming in, so that's great. Maybe I'll give a couple more minutes just in case some of you guys are still wrapping up. Great. So I'm seeing a lot more done twos now. So I'll move on and to the answers for this one. And thank you guys all for the done twos. I really appreciate that. It really lets me know that you're on track and not leaving too many people behind. So hopefully you were able to come up with something similar to what I have up here, which is that the cluster up here, the olive cluster, was a B cell cluster, whereas this blue cluster is a cluster of monocytes. So now that we've figured out what these clusters are that go up in patient 13 as they recover, let's do a little bit of interpretation. So the cluster 12 that I annotated, this was red blood cells. And these are a cell type that are actively excluded from PBMCs. So this likely just represents, a little bit of overzealous pipetting. This is not something biologically interesting. The next cluster, the olive-colored cluster 7 up here, was a cluster of B cells. And this particular B cell cluster expresses high levels of IgG antibody genes. You might have had a chance to look into this if you finished early and did the bonus exercise where you were comparing to cluster number 2. And that cluster number 2, it's also a B cell cluster, but it has high levels of immunoglobulin M antibody genes. And the fact that this cluster 7 goes up in abundance with recovery, it's consistent with what we know about the course of a typical COVID-19 infection, where immunoglobulin G antibody levels, they rise later on in the course of infection. The last cluster that we looked at was this cluster 4, which was CD14 monocytes. And this same observation was made by the authors that reported this data, that this cluster went up in patient 13 as they recovered. And they went on to characterize a much larger set of samples in this particular cell type. And so what we're going to do for our next exercise is we're going to make a plot just to make it very clear how this cluster is changing between our samples. This plot here, it's not the best for evaluating abundance shifts between your samples because it doesn't account for sample size differences. So we have a lot fewer cells in my blue sample here. So we're going to make this more clear. And to do that, we're going to use the plots and tables module. So as its name suggests, this is where you can access a number of standard plots for your data set. Importantly, you can fully customize the styling of these plots to hopefully get it to be acceptable for a publication or for a presentation. And you can obviously export these plots for those purposes as well. So we're focusing on these CD14 monocytes for this exercise. And to do this, we're going to create two custom cell sets. The first is we're just going to duplicate the CD14 monocyte cluster. So to do this, you're going to go and select cluster 4, and then you're going to click this second button up here. And that's going to create a new cell set by combination. So in this case, we've just selected one set of cells, so it's just going to duplicate that cell set. Give it an appropriate name, CD14 mono, and then still with that cluster 4 selected, come up and click this last button up here, and that's going to create a new cell set by complement. So it's going to be a cell set from using all cells other than the ones that you have selected. So give that an appropriate name, all other cells. Once you've done that, come down to the plots and tables, open up a frequency plot, and then under the select data heading, group your data by sample. And then for the second drop down here, group your data by custom cell sets. Those are the cell sets that you've just created. Take a look at that, and when you're done, please give me a big done 3 in the chat. And this should just take you guys a few minutes.
So while everybody's working on the third exercise, I just have a question. So I've logged in a couple of the dummy accounts and also my account from last year. And under data, Alex, I flagged this to you, data processing. I'm not able to see any of the plots. Step 6 or 7, I can see plots under plots and tables, but I'm just curious to wonder if any of the other folks who we gave dummy accounts to had the same issue. Okay.
Yeah, definitely let us know if you're experiencing what Jacqueline is experiencing. I'm not exactly sure what's going on, but I'll definitely take a look at it after. But yeah, let us know if you're seeing the same thing. And it's really great to see all of you putting the done 3s down there. Very helpful. I really appreciate it. And definitely let us know if you have any questions as well. It's really great to be getting questions from you guys.
As the meaning of your name. I am a photographer.
Great. So I'm seeing a lot of done threes in the chat. So I'm going to move on just because I'm slightly unclear how much time I'm going to be able to, whether I'll be able to finish up here. So I'll move on. So hopefully you were all able to make a plot that looks similar to this one, where we can see clearly that our two acute samples, and particularly our sample from patient 13, has a lower proportion of those CD14 monocytes in comparison to the sample that was collected from patient 13 during the convalescent phase. So this would be, reasonable evidence to want to follow up with this set of cells and see what else is different about them. And that's exactly what we're going to do for our next exercise, where we're going to perform a differential expression analysis between our acute and convalescent samples. And so in order to do this, we're going to reuse those two cell sets that we created in the last exercise. So the first step is just going to be to come up to the differential expression tab up here. And this time you're going to select that second radio button. So that's the one you want to select when you're doing comparisons between groups of samples. And you're going to compare the P13 convalescent sample in one of those drop downs, and you're going to compare it to the rest of the samples, so the other two samples.
I'm going to ask him. Alex, sorry, we have a question that came to just one of us.
Sure.
Which is removed. Oh, I see. What if you have more than one custom cell set, I believe was the question.
So, I mean, yeah, if for this particular data set, if you created more than, one, those two cell sets, obviously your plot would not show up correctly. Yeah, unfortunately, I think what the question is getting at is whether or not you can just like create a different custom cell sets grouping. And you can't do that now. I've heard a few people requesting it. So it's definitely something on the radar for, a possible future feature. But right now there's just the one custom cell sets grouping. But great question. After you've selected that second radio button and selected the samples that you're going to compare, you're also going to select the cluster 4 to run that comparison. Run that differential expression analysis, and that's going to give you a list of differentially expressed genes. And the first thing we're going to do is filter those genes to only have the ones that are significant. So you're going to go click Advanced Filtering, and under Presets, select Significant, click OK, and that's going to filter your table so that you're only looking at genes that have an adjusted p-value less than 0.05. The next thing that we want to do is we want to put all those genes into our heat map. So to do that, you're going to come and click this little checkbox here to select all the genes in your table, and then you're going to click Heat Map and select Overwrite. And it might take a few seconds to grab all the data, but that's going to put those genes over in your heat map. If you've gotten that far, you're doing good so far. The next step is that we want to get this samples metadata track on our heat map. So to do that, we're just going to click this cog, and we're going to toggle on the samples metadata track. And once you've done that, the last step is just to come up to your custom cell sets and hide the all other cell sets that you previously created. And that's going to make it so that you just see this, you know, your cluster 4, and also so that you're just looking at the expression for cluster 4. Once you've done that, take a look at these few sentences, see what you make of them, and then give me a big done for in the chat, and I'll give you guys about 10 minutes for this exercise. I probably won't give you guys 10 minutes for this exercise because that's going to bring us to the end of today, but I'll give you guys a few minutes to get started on this. And if you have any questions, ask them, but we'll probably leave this for homework. We have a question in the chat. Martin was wondering about trajectory analysis on the plot section, whether it's also under development. Yeah, I would definitely say it's, you know, there's additional features that should definitely be added to that. It's fairly rudimentary right now. You can, you know, you can get those trajectories as Peter described. It's missing some features where often for trajectory analysis, what you're interested in is what genes are changing along a trajectory. And that's not something that you can do right now. So yeah, that's definitely, you know, there's a great thing to be adding to it, but yeah, it's definitely things that need to be added. Great, so I'll give you guys a couple more minutes, and then I'll move along just for time's sake. Okay, so I'm gonna move along. So I apologize for not giving you guys enough time to finish off that exercise. There's been a lot of really great interactivity in this workshop, which I really appreciate, and hopefully you will take another chance at this exercise on your own time. I think it's a really great exercise for showing just how adaptable your explorations of your data set can be. So hopefully you'll take a look at that again. In terms of the few sentences up here in the top, we can see that one of these genes, FCGR1A, which is the IgG receptor, we can see that it's consistently upregulated in our two acute samples and that it's downregulated in the convalescent sample. And this same finding was reported by the authors that first collected this data. The other gene that I got you guys to look at, or you might look at later on, is RNase 1. And this wasn't reported by the authors, but it shows a similar pattern where it's up in the acute phase and then it goes back down with recovery. And this was just something that I thought was interesting, given that RNase 1 is an RNA degrading enzyme. So it could represent part of the antiviral response. And it would make sense that it comes back down after recovery. So that's the end of the exercise portion of the workshop. And all that's really left for me to do is to make sure that you know how to upload your own data to Cellenics so that you can analyze your own data set. So hopefully it's fairly straightforward in terms of the UI where you do that. You're going to go to data management and then click the create new project button. Once you've given your project a name, then you need to add data to your project. And I'll go over in the next slide exactly what format that data needs to be in. But before I do, I'll also just mention that this is where you want to add metadata for your samples. So if you click this button, it's going to create a new column in your table, and you're going to use that for indicating groupings of your sample. So we could have had a column that said time point, that titled it time point and made it acute for the first two samples and convalescent for the last sample. We could have had another column that said patient ID and made it P14 for the first sample and P13 for the second and third samples. And this is really important to do because you're going to be able to access these metadata groupings downstream in the platform. So you're going to be able to use them for your plots. You're going to be able to use them to run your differential expression analyses. So definitely do make sure you add those if you have a multi-sample data set. So we've got a question in the chat. Slater is asking, is this how we would identify multiplex data? So great question. So Cellenics doesn't currently support uploading multiplex data. So you have to demultiplex it first. If you need any help with that, please get in touch. I'm more than happy to help or to direct you to a script if you want to take it on yourself. Great. So in terms of the data formats that Cellenics does support, you can currently upload data that's been generated either with 10x Chromium, BD Rhapsody, as well as data sets that have been pre-processed with Seurat. So these would be saved as RDS files. In terms of what I'm going to focus on for today, I'm just going to focus on 10x Chromium because this is going to be what most of you are uploading to the platform. So you're going to be uploading either the filtered or the unfiltered count matrices that are output from Cell Ranger. And usually, if you're getting the data from a core, you'll find these inside of a folder called raw feature BC matrix. And you're going to need 3 files for each sample. So you'll need a features.tsv file, you'll need a barcodes.tsv file, as well as a matrix.mtx file. And these can be gzipped or not, it doesn't matter. So you take these 3 files for your sample and you put them into a folder with the sample name. So here we have sample 1 and we have those 3 files. And you want to make sure that these 3 files don't also include the sample name. Otherwise, Cellenics won't recognize them. Once you've done that for all of your samples, you're just going to take these folders and then you can drag them into the platform. So you're going to click that add data button, and then you can drag them into the pop-up that comes up. And yeah, that's it for the workshop. So just summing up, I really hope that, you know, as a result of this workshop, you understand a little bit more about how you can use Cellenics for your single cell analysis, that you're comfortable using it to explore a data set in Cellenics, and most importantly, that you know how to get help if you need any help using Cellenics. I'd love to hear about the projects that you're working on, so do definitely, you know, get in touch with me by email or just book a Zoom with me. Thank you all again for the great interactivity. There's lots of great questions and all the done for in the chat. And I'm more than happy to answer any other questions that you guys have now. I don't mind staying an extra 5-10 minutes after. So if there's any questions, definitely speak up.
Thank you all. And just want to say, remind you again to check the CCB website for regular updates on Cellenics, and keep an eye out for more info sessions, seminars, and things, workshops like this one. And also we are starting like an annual outreach to users where we're giving our users an opportunity to request different features or updates that they would like to the platform. So we're here to support you, and we look forward to helping you.
Great. Thanks, Jacqueline. And there's a couple questions in the chat, so I'll answer those as well.
Okay, I've got to jump to another call, but thanks, Alex.
Okay, great. Thanks, Jacqueline. Take care.
Yeah.
So Yang King is asking, is there a way to export intermediate data after done with data processing step? For example, to get the cluster information. So great question. Unfortunately, it's not as good as I would like it to be, but it's something that will be updated soon. You can export your data by going to data management and clicking this download button, and you can download the process SURAT object. So that's after data processing. The one issue with this right now is that, as you mentioned, it doesn't have that cluster information. This is sort of, an unfortunate side effect where we're storing that as a different file in the platform. But it's something that is, this feature has already been added. It just needs to be released. So, within a month, you will be able to do that. And that will apply for any previous data sets that you've created as well. So a great question. That will definitely be there soon. And then Martin is also asking, there is an action name, calculate pseudo time under trajectory analysis. It seems to me it is disabled or it needs special instruction. So great question. I'll show you how to use that feature just in case you need to use it. So the first thing you need to do is calculate root nodes. That will, take a little bit of time. I'm assuming you've already done that, given the barrier that you're coming across. After you do that, you then need to select your root node. So there's some interactivity to the plot. You can click on it to select nodes. And then after you select those nodes where you want to, start your trajectories, then you should be able to click the calculate pseudotime button. And then, Carolina is asking, when do you think the recording of the session will be available? It should hopefully, I don't know for sure, but should be available soon.
I just
Probably
put a
next
comment
week.
that I'll try to put it in the on the website the next couple of days.
Okay, great. Thanks, Jenna. So Pamela is asking, is there a cost for Harvard affiliates? No, there's no current cost for using Cellenics for Harvard affiliates. And I'm not seeing any more questions. So again, thank you to everybody who's still here, and definitely get in touch if you need any help.